Hi. So this is the vagina. Welcome to the vagina with Stephen Sparks. Alright, so the vagina is the fibromuscular tube of the female reproductive tract that leads to the exterior of the body, if you didn't already know. The wall of the vagina consists of three layers: a mucosa, a muscularis, and an adventitia. The epithelium of the mucosa is non-keratinized squamous, stratified squamous. It undergoes changes that correspond to the ovarian cycle. The amount of glycogen stored in the epithelial cells increases under the influence of estrogen, whereas the rate of desquamation increases under the influence of progesterone. The glycogen liberated from the desquamated cells is fermented by Lactobacilli vaginalis, producing lactic acid that acidifies the vaginal surface and inhibits colonization by yeasts and potentially harmful bacteria. The vagina has certain histologic similarities to the proximal portion of the alimentary canal, but is distinguished by the following features. The epithelium does not keratinize, and except for the deepest layers, the cells appear to be empty and routine H&E stains. So you can kind of see that here. The mucosa contains neither glands nor muscularis mucosa, and the muscle is smooth and not well ordered. This should be contrasted with the oral cavity, pharynx, and upper part of the esophagus. We don't care about that right now. Okay, so this picture. The mucosa of the vagina consists of a stratified squamous epithelium, stratified squamous epithelium, and underlying and ununderlying fibrous connective tissue, that appears more cellular than other fibrous connective tissue. The boundary between the two is readily identified because of the conspicuous staining of the closely packed small cells of the basal layer of the epithelium. So this right here. Connective tissue papillae project into the underside of the epithelium, giving it the epithelial connective tissue junction uneven appearance. The papillae must be or may be cut obliquely or in cross section, and thus it can appear as islands sometimes right here. The epithelium is characteristically thick, and although keratinohyaline granules may be found in the superficial cells, keratinization does not occur in the human vaginal epithelium. Thus, nuclei can be observed throughout the entire thickness of the epithelium, despite the fact that the cytoplasm of most of the cells above the basal layers appears empty. These cells are normally filled with large deposits of glycogen that is lost in the fixation process of these tissues. That's why you don't see it on these stains and stuff, or in these pictures. The rectangle outlines a portion of the epithelium and connective tissue papillae that is examined at higher magnification below, right here. The muscularis layer of the vaginal wall consists of a smooth muscle arranged in two ill-defined layers. The outer layer is said to be generally longitudinally arranged, and then the inner layer is circularly arranged. But the fibers are usually organized as interlacing bundles surrounding by connective tissue, so you can't really tell a clear difference. Many blood vessels are seen in the connective tissue, so it's good to know. Right here, 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 here. All right, what else can we learn? So, in this picture right here, this is a higher magnification of the epithelium. The obliquely cut cross-section portions of the connective tissue papillae that appear as connective tissue islands in the epithelium are more clearly seen here, right there. In some instances outlined by the surrounding closely packed cells of the basal epithelial cell layer. Note again that the epithelial cells, even at the surface, still retain the nuclei, and there's no evidence of keratinization. So at this point I can ask a practice question. Is the vaginal epithelium keratinized? No, it is not. Okay. This bottom left picture. This is a higher magnification micrograph of the basal portion of the epithelium between the connective tissue and papillae. Note the regularity and dense packing of the basal epithelial cells. Okay, boom, 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 right here. They are the stem cells for the stratified squamous epithelium. Just like every other thing we've ever learned, you always have the stem cells at the bottom, and then they work their way up as they, whatever. The outer cells of these cells migrate towards the surface and begin to accumulate glycogen as they go, and become less regularly arranged as they go as well. The highly cellular connective tissue immediately beneath the basal layer of the epithelium typically contains many lymphocytes. Hey, lymphocytes. The number of lymphocytes varies with the stage of the ovarian cycle. Lymphocytes invade the epithelium around the time of menstruation and appear along the epithelial cells and vaginal smears. So that makes sense, right? Because you're starting to slough things off, so there might be access of bacteria to layers that it wouldn't otherwise be exposed. I don't know if that makes sense, actually, because you're not sloughing off the vagina when you menstruate. But anyway, who cares? Moving on. Just know at the time of menses, there's more lymphocytes there, I guess. Okay, so on the right-hand side, we've got the muscularis vagina. So this higher magnification micrograph of the smooth muscle of the vaginal wall emphasizes the irregularity of the arrangement of the muscle bundles. So at the right edge of the figure is the bundle of smooth muscle cut in longitudinal section, and adjacent is the circular section. This bundle abuts on a longitudinally sectioned lymphatic vessel. To the left of the lymphatic vessel is another longitudinal bundle of smooth muscle. A valve is seen in the lymphatic vessel, and a small vein is seen right here. Isn't that cute? Okay, that was all that fun stuff. So now let's check out the breast tissue. All right. Mammary gland inactive stage. Inactive mammaries. So the mammary glands are branched tubulo-alveolar glands that develop from epidermis and come to lie in the subcutaneous tissue. All right, the superficial fascia. They begin to develop at puberty in the female, but do not reach fully functional state until after pregnancy. The glands also develop in the male at puberty. The development is limited, however, and the glands usually remain in a stabilized state. So this first picture is a section through an inactive gland. The parenchyma is sparse and consists mainly of duct elements. Several ducts are shown in the center of the field. A small lumen can be seen in each, and the ducts are surrounded by loose connective tissue. Okay. Yeah. Together, the ducts and surrounding connective tissue constitute a lobule. Two terminal duct lobules, like here and here, are called these are called terminal ductal lobule units. So two terminal ductal lobule units, TDLU, are bracketed in this picture. Beyond the lobular unit, the connective tissue is more dense. So out here it's more dense compared to, say, around here. Okay. All right. Now, let's take a look at this next picture. All right, higher magnification. In distinguishing between the loose and dense connective tissue, recall that both extracellular and cellular features show differences that are both evident in the figure at the inset. Note the thicker collagenous fibers in the dense connective tissue, in contrast with the much thinner fibers of the loose connective tissue. So you can just tell the difference between this and this. The loose connective tissue contains far more cells per unit area and a greater variety type of cells. This figure shows clusters of lymphocytes right here. And in the inset, you can actually see plasma cells here and here, and individual lymphocytes. Okay, both plasma cells and lymphocytes are cells with a rounded shape, but the plasma cells are larger and show more cytoplasm. In addition, the regions of plasma cell cytoplasm display basophilia. Elongate nuclei and spindle-shaped cells belongs to fibroblasts. In contrast, although cell types in the dense connective tissue may also be diverse, a simple examination of equal areas of loose and dense connective tissue will by far show fewer cells in the dense connective tissue. Characteristically, dense connective tissue contains numerous aggregates of adipocytes, like this here. The epithelial cells within the resting lobular units are regarded as being chiefly duct elements. Usually alveoli are not found. Their precursors, however, are represented as cellular thickenings of the duct wall. The epithelium of the resting lobule is cuboidal. In addition, myoepithelial cells are present. If you look at the inset, shows a thickening of epithelium in one location, presumably a precursor of an alveolus, and myoepithelial cells at the base of the epithelium. As elsewhere, the myoepithelial cells are on the epithelial side of the basement membrane. During pregnancy, the glands begin to proliferate. This can be thought of as a dual process in which ducts proliferate and alveoli grow from the ducts. All right, so that was inactive breast tissue. Okay, inactive breast tissue. All right, now let's talk about the mammary gland during the late proliferative and lactating stages. So, wow, that looks different. So much different. Okay. All right. Mammary glands exhibit a number of changes during pregnancy and preparation for lactation. Lymphocytes and plasma cells infiltrate the loose connective tissue as the glandular tissue develops. As the cells of the glandular portion proliferate by mitotic division, the ducts branch and alveoli begin to develop at their growing ends. Alveolar development becomes most prominent in the later stages of pregnancy, and the accumulation of secretory product takes place in the alveoli. At the same time, lymphocytes and plasma cells become prominent in the loose connective tissue of the developing lobules. Myoepithelial cells proliferate between the base of the epithelial cells and the basal lamina in both the alveolar and the ductal portion of the glands. They're most prominent in the larger ducts. Both merocrine and apocrine secretion are involved in the production of milk. The protein component is synthesized, concentrated, and secreted by exocytosis in a manner similar for typical protein secretion. The lipid component begins as droplets in the cytoplasm that coalesce into large droplets in the apical cytoplasm of the alveolar cells and cause the apical plasma membrane to bulge into the alveolar lumen. The droplets are surrounded by a thin layer of cytoplasm and are enveloped in plasma membrane as they are released. The initial secretion of the first days after birth is called colostrum. This pre-milk is an alkaline secretion with a higher protein, vitamin A, sodium, and chloride content than milk, and a lower lipid, carbohydrate, and potassium content. Considerable amounts of antibodies are contained in colostrum, and these provide the newborn with passive immunity to many antigens. The antibodies are produced by the plasma cells in the stroma of the breast and are carried across the glandular cells in a manner similar to that for secretory IgA in the salivary glands and intestine. So a few days after parturition, the secretion of colostrum stops and lipid-rich milk is produced. So let's take a picture or look at this picture. So whereas the development of the duct elements in the mammary gland occurs during the early proliferative stage, the development of the alveolar elements becomes conspicuous in the late proliferative stage. This figure shows the terminal duct lobular units at the late proliferative stage. So here, here, here. Individual lobular units are separated by narrow, dense connective tissue septa. Okay. The connective tissue within the lobular unit is a typical loose connective tissue that is now more cellular, containing mostly plasma cells and lymphocytes. The alveoli are well developed and many exhibit precipitated secretory product. Each of the alveoli is joined to a duct, although their relationship can be difficult to identify. The epithelium of the intralobular ducts is similar in appearance to the alveolar epithelium. The cells of both components are secretory, so the alveoli as well as the intralobular ducts consist of a single layer of cuboidal epithelial cells subtended by myoepithelial cells. Often what appear to be several alveoli are seen merging together with one another, like this right here. Such profiles represent alveolar units opening into a duct. So interlobular ducts here are easy to identify as they are surrounded by dense connective tissue. In one instance, an intralobular duct can be seen emptying into an interlobular duct. Again here. So this is the intralobular duct. This is the interlobular duct it's emptying into. The inset shows secretory epithelium in a much higher magnification. Note this is simple columnar epithelium. The nucleus of a myoepithelial cell. Oh, look at that. Isn't that cool? It's seen at the base of the epithelium. Generally, these cells are difficult to recognize. So, yeah, good stuff. That was that picture. Let's look at this last picture. You guys are probably tired of hearing my voice. The specimen shown here is from a lactating mammary gland. Look at that. That's cool, huh? It is similar in appearance to the gland at the late proliferative stages, but differs mainly to the extent that the alveoli are more uniform in appearance and their lumina are larger. As in the late proliferative stage, several alveoli can be seen merging into one another. Right here. Where there's an asterisk, you can kind of see that, right? The use of osmium in the specimen stains the lipid components of the secretion, so the inset reveals the lipid droplets within the epithelial cell cytoplasm, as well as lipid that has been secreted into the lumen of the alveolus. The lipid first appears as small droplets within the epithelial cells. These droplets become larger and ultimately are secreted into the alveolar lumen along with milk proteins. The milk proteins are present in small vacuoles in the apical part of the cell but cannot be seen by light microscopic methods. They are secreted by exocytosis. The lipid droplets, in contrast, are large and surrounded by the apical membrane as they are pinched off to enter the lumen. Thus, it is an apocrine secretion. Several interlobular ducts are evident in this picture by D, the letter D. One of these ducts reveals a small branch, an ending intralobular duct joins the interlobular duct here. That, my friends, was the end of that section. So this is just the plates in the back of Ross that I'm showing you, just to kind of, so that when you're going through doing his resource, or you're going through and doing his practice questions or whatever, you can look and see, you kind of recognize where you're at least that much, you know, and then I'm sure the details and stuff you need to get from his resources as far as the function of a lot of these things, but these are the basic bare bones how to identify these tissues that we're supposed to identify this phase. Yeah, good luck.